Welcome back, everyone. We are talking about signal transduction, and in our last video, we talked about how an action potential is created and how it's propagated down a nerve cell. And now we've gotten to the end of this nerve cell, and it needs to be propagated to the next cell. So when two nerve cells are communicating with each other, this area is called a synapse. And there's a couple of different kinds of synapses that can happen. One is an electrical synapse. And in here, your presynaptic neuron, the one that has the message, is connected to your second neuron, the postsynaptic neuron, where you're going to send the message. They're connected by gap junctions, these little pores that connect the two cells and allow ions to be transferred directly from one cell to the next. So the ion will move through the cell junction. The sodium can move straight through there. There's no delay in transmission. It's almost the same as just continuing down through the same cell. You just continue your action potential, and the next cell will be excited. There are also chemical synapses. And in a chemical synapse, your presynaptic cell here and your postsynaptic cell here, there's no gap junction between them. There's a little space between them. And this little space is called a synaptic cleft. And you're going to have to be able to get that message from your presynaptic cell to your postsynaptic cell without ions actually flowing through there. You're going to have some chemical from the first cell send a message to the second cell. The chemical that is used is a neurotransmitter. Those neurotransmitters are stored in little vesicles here at the end of the synaptic baton. And when the action potential arrives down that axon, that is going to trigger the excitatosis of these neurotransmitters, and they'll just diffuse through the cleft and attach to receptors in the postsynaptic cell. Then when those neurotransmitters bind to the cell, bind to those receptors, that's going to create an electrical signal to continue the action potential in the receiving cell. There's two kinds of neurotransmitter receptors. Some are ligand-gated ion channels, or sometimes you'll hear them called ionotropic receptors. And in here, the neurotransmitter binds to the receptor, and it's a ligand-gated ion channel. So the channel just opens right up, and the ions can flow in and out, allowing the action potential to be propagated. There are also some that are a little bit more indirect. They're called metabotropic receptors. So when the neurotransmitter binds to the receptor, it is going to activate a signaling cascade that results in an ion channel being opened. So while they both end up opening an ion channel to propagate that action potential, they're doing it in very different ways. Right here, the ligand opens a channel. Here, the ligand starts a messaging system, and that relay is going to open the channel. A neurotransmitter is any signaling molecule that's released by that presynaptic neuron, and then they'll be mostly detected by the postsynaptic neuron with the receptor. Some receptors will be excitatory. That means it will cause depolarization of the postsynaptic cell and activate or excite that cell. And some can be inhibitory. When a neurotransmitter binds to an inhibitory receptor, it's going to cause the postsynaptic cell to hyperpolarize or go further away from reaching that action potential. Here's some different types of neurotransmitters that our cells use. Acetylcholine, which is excitatory to vertebrate skeletal muscles, and it can be excitatory or inhibitory at other sites. Norepinephrine, which is excitatory or inhibitory to both the central nervous system and the peripheral nervous system. Dopamine, which is generally going to be excitatory, but it could be inhibitory. GABA, or aminobutyric acid, which is inhibitory for your central nervous system. Glutamate, which is excitatory to the central nervous system. Glycine, which is inhibitory to the central nervous system. And serotonin, which is generally inhibitory to the central nervous system. Then some neuropeptides, we have substance P, which is excitatory to both the central and peripheral nervous system. This endorphin is generally inhibitory to the central nervous system. Anandamide is inhibitory to the central nervous system. And nitric oxide, which could be excitatory or inhibitory, used by the peripheral nervous system. Acetylcholine, this first one, is the most common neurotransmitter in vertebrates. It's in synapses outside the central nervous system, and also for neuromuscular junctions, or those synapses where a neuron signals to a muscle. It's an excitatory neurotransmitter, and a synapse that uses acetylcholine as the neurotransmitter are called cholinergic synapses. Neurotransmitter secretion is going to be directly controlled by the concentration of calcium ions in that synaptic baton, and it's that increase of calcium that is going to trigger the exocytosis of these vacuoles here. And when an action potential arrives, so you've got this action potential coming down the axon, it's going to open voltage-gated calcium channels, like this channel here, which allows calcium to enter. And the neurotransmitters are going to be stored in these neurosecretory vesicles here in the baton. And when calcium is released in the baton, that's going to do two things. It's going to allow these vesicles to be mobilized so they can release. And vesicles that are already ready for their release will be able to dock and fuse with the cell membrane to allow exocytosis of that neurotransmitter into the synaptic baton region. The neurotransmitters will be secreted from the cell through exocytosis. In this process, the vessel is going to fuse with the plasma membrane. So here you can see that fusion happening. The arrival of the action potential at the axon terminal is going to open the voltage-gated calcium channels. Calcium enters the synaptic baton. And vesicle fusion is going to involve those vesicles that are already docked at the plasma membrane. And this is going to use our T snare and V snare proteins, just like our other exocytosis discussions we've had. And calcium in the batons is bound to synaptotagmin, which is going to have a change in its conformation. That's going to allow the T snares on the plasma membrane and the V snares on the vesicle to interact and allow those vesicles to merge into the plasma membrane and exocytose their contents. Docking is going to happen at the active zone. That's where the synaptic vesicles and calcium channels, you can see they're very close together here. There are two illnesses, tetanus and botulism, that can interfere with docking and release. These two bacteria, the bacteria that cause tetanus and botulism, release a neurotoxin that blocks that. Tetanus is sometimes called lockjaw because it forces all of your muscles to contract, because by blocking that neurotransmitter signal, your muscles are not able to relax. And botulism kind of works in an opposite way, that it'll force all of your muscles to kind of relax and droop. You can't even like open your eyelids for that. So those two are able to control your muscle movement because they block that docking and release of your neurotransmitters to change and control your muscles. To keep the size of the nerve terminal the right size, there will be compensatory endocytosis. That means we're just going to pull some of that plasma membrane back into the cell just to bring it back in and maintain the proper balance of membrane in that cell. All of those different neurotransmitters have different receptors that will detect them and bind to them. Acetylcholine will bind to a ligand-gated sodium channel, so it'll trigger the opening of those sodium channels. And this receptor is called the nicotinic acetylcholine receptor. And when two molecules of acetylcholine are going to bind, it lets the receptor open, and sodium here now can rush into the cell. Then, now that you have the neurotransmitter in the synaptic cleft, you've allowed that signaling to be propagated to the next cell. If you leave that neurotransmitter there, that signal is going to keep being received by that postsynaptic cell. And we don't want to do that. We want to send the signal and then stop the signal. So we have to rapidly remove that neurotransmitter after it's been sent. There's two different ways that cells can do that. One is neurotransmitters can be degraded. So once it's been released, it breaks down. Another is that the neurotransmitter can be reuptaken. So that means that that neurotransmitter is going to be pumped back into the presynaptic cell. It can be put back into vessels or recycled somehow. Or it could be put into nearby support cells. And this reuptake, you may have heard of selective serotonin reuptake inhibitors, a type of antidepressant drug, sometimes called Prozac. It blocks the reuptake of neurotransmitters such as serotonin, allowing serotonin to still be there and send that signaling as a treatment for depression. And then usually in a cell, one action potential is not enough to trigger a postsynaptic cell to respond. You're going to need to have something more than just the one. When you have incremental changes in potential from a neurotransmitter, that's called a postsynaptic potential. This can be excitatory, causing the receiving cell to be excited, or inhibitory, causing the receiving cell to be inhibited. That's going to depend on what transmitter and receptor is involved. So to have enough of a strength to actually cause that postsynaptic cell to respond, you're going to do a couple of different things. One could be temporal summation. So that means that you have two action potentials in rapid succession. So that one by itself is not enough. It didn't hit that threshold and the cell is not going to respond. But when you have this temporal summation, temporal meaning time and summation meaning adding, just like, the sum for adding. So you have one and then another, and that hits the threshold by having these two signals very rapidly together. So the cell can depolarize. And so rapidly having EPSPs over time will keep that cell past its threshold and allow the cell to respond. There's also spatial summation, which means that you have several signals coming from different spaces or from different cells. So all of these action potentials together are going to add together to trigger that receiving cell to respond and induce an action potential. By having many action potentials releasing neurotransmitter from different cells simultaneously, it's more likely to get that action potential. So this is spatial summation. So you can have more than one action potential at the same time from different spaces, from different cells, which is spatial summation. Or you can have multiple from the same sending cell, the same presynaptic cell that just keeps sending multiple signals over time, quickly over time. And that's temporal summation. Okay, I hope you've enjoyed our discussion of our cell signaling mechanisms. We're going to come back in our next unit. We're going to talk about genetics. We're going to talk about transcription and translation, how we use our genes to make proteins. We're going to talk about how we control our genes, how some genes will be turned on in some cells, some will be turned off, how they can be modified. So we have a pretty exciting end to this semester. I will see you soon. Bye.